So next subtopic is principle of immunofluorescence. So we will see how this immunofluorescence works. We know light is an electromagnetic radiation that is detected by human eye, and the radiations are present in the light. They range from 400 to 700 nanometer in wavelength. Fluorophores absorb light of a specific wavelength, then they get excited. They change from low energy state to high energy state, and when the electrons they change to high energy state, they come back and then they release energy, and this energy is visible when we use fluorescent microscope. So fluorophores absorb light of a specific wavelength, they get excited and then emit the light of a different wavelength, which is visible, which is seen in the fluorescent microscope. Fluorescence is a result of light capturing ability of fluorophore molecules and then its emission, which is detected. And fluorescent materials give off distinct color when they are exposed to different sources of light. Here we can see that once the fluorophore absorb light, which is shown in blue color, they get excited, and these excited molecules, when they come back to their original state, they release energy, and this is then may be seen in the form of emission or emission spectra, or light which is emitted, which is in the form of red color. So once the fluorophore is exposed to different type of lights, they get excited, they absorb light, they get excited, and then the energy is released, and this energy which is released, that is visible in the fluorescent microscope, with the help of which we can see our samples. Immunofluorescence uses specificity of antibodies to the respective antigens in the cells or the tissues. So by using this specificity, we are able to see the respective antigens in cells and tissues. And immunofluorescence makes use of fluorophores to detect the location of antibodies and, of course, their respective antigens. Normally, fixed cells or tissues, they are used because antibodies, they cannot pass through the membrane of the cells. These are larger molecules, and that's why these antibodies, along with their fluorophores, they cannot pass through the cell membrane of the cells. So that's why normally fixed cells or tissues, they are used. And before using these immunofluorescent labeled antibodies, we permeabilize the cells. We make the pores in the cell membrane of the cells so that these antibodies can enter the cells, and in this way they can stain the antigens. But in some cases, living cells can also be used. So proteins in the supernatant or on the outside of the cell membrane can be bound by the antibodies, and in this way this allows also that living cells can be stained by using fluorescent microscope.